Hello, my name is Sam Sloan, and this is a presentation regarding my research in coordination with the Graham Lab and the Alameda County Department of Public Health concerning carbapenem-resistant Enterobacteriaceae, specifically E. coli from Alameda County, and subsequent whole genome sequencing assembly and characterization. Ever since their discovery, beta-lactam antibiotics, such as penicillin, have been used to treat gram-negative bacterial infections. Cephalosporins are a branch of beta-lactams, which were added to the antibiotic arsenal in 1964. They function through targeting enzymes, which are involved in the cross-linking of the bacterial cell wall. In the 1990s, Enterobacteriaceae, a family of gram-negative bacteria, began to develop resistance to cephalosporins through the acquisition of extended-spectrum beta-lactamases, otherwise known as ESBLs. These enzymes are able to hydrolyze the beta-lactam antibiotics, rendering them ineffective. To date, beta-lactam production remains the most important contributing factor to beta-lactam resistance. As you might surmise, another form of beta-lactam antibiotic was discovered. This form was called carbapenems. They are one of the few remaining antibiotics that can treat ESPL-producing bacteria. But as you also might surmise, a resistance enzyme called carbapenemase that makes carbapenems, penicillins, and cephalosporins ineffective is on the rise, limiting the treatment options, if any, to treat these infections. Carbapenem-resistant Enterobacteriaceae is an urgent public health problem worldwide. These multi-drug resistant organisms exhibit resistance to most, if not all, available antibiotics today and are associated with considerable mortality. In the US alone, in 2017, there were 13,000 infections with 1,100 deaths, relating to $130 million in total associated healthcare costs. The World Health Organization considers CRE such a high threat that in their most recent publication of priority pathogens, they've listed carbapenem resistance Enterobacteriaceae as critical, their highest level. These organisms are critical not only due to their disease burden, but because of the disease burden potential. This increased potential comes through the spread of resistance through horizontal gene transfer of mobile genetic elements. This is the process through which one organism can transfer genetic material to another, and these organisms are able to transfer resistance. The Enterobacteriaceae family is broad and diverse, containing members such as Enterobacter and Klebsiella, among many others. However, in this study, we chose to specifically focus on Escherichia coli, one of its dominant pathotypes, extraintestinal pathogenic E. coli, otherwise known as XPEX. XPEX are a broad collection of pathotypes that are facultative pathogens colonizing the extraintestinal environment of hosts. XPEX are the most common gram-negative bacterial pathogens in humans, accounting for the largest majority of UTIs, bacteremias, and sepsis globally, and currently increasing in mortality and morbidity. XPEX are derived from a small number of phylogenetic lineages, many of which are considered pandemic lineages. 4 sequence types, defined using multi-locus sequence typing, are responsible for nearly half of all E. coli urinary tract and bloodstream infections in the world. These are ST131, 69, 95, and 73. Before 2000, XPEX were mostly susceptible to first-line antibiotics. However, as the use of beta-lactam antibiotics to treat XPEX increased, the predominance of highly resistant XPEX, otherwise known as ESBL XPEX, due to their acquisition of the extended-spectrum beta-lactamase genes, were now widespread and an important cause of UTI and bacteremia. Which brings us to our study. The Alameda County Department of Health has a public health mandate that requires any carbapenem-resistant samples be sent to their public health lab for further analysis. They come from a broad set of infections and are such important organisms that they must be sent to identify if there's an outbreak in CRE. However, our research is intended to discern if there's biological factors relating to these organisms and their infections. The Alameda County Public Health Laboratory provided our lab with Illumina unpaired unidirectional sequence reads from their in-house library curation and whole genome sequencing of 82 carbapenem-resistant E. coli isolates. Whole genome sequencing is a method used to reconstruct the genome of an organism in one process. From these genes, I hope to identify and characterize their phylogenetic relationships and sequence types, as well as identify antibiotic resistance genes and virulence factor genes, and evaluate for relationships between sequence types and genes. First, we assembled the short read sequences using Unicycler. We trimmed the short reads, a function for quality, and then generated a consensus sequence through de novo assembly. De novo assembly is a method to assemble the genome for which you have no reference. This assembly was then checked for completeness of coverage by mapping the reference against the consensus sequence. I then used in silico Claremont phylotyping to identify phylogroups based upon conserved gene sequences. Furthermore, multilocus sequence typing was used to identify sequence type based on differences of a different set of conserved genes. And then finally, FIMH typing was also performed by evaluating the genetic sequence of the FIMH gene within the genetic sequences. The FIMH typing is a method used to evaluate the relationship between organisms of the same ST type. We used Abricate to analyze the results for the prevalence of antimicrobial resistance genes and virulence factor genes. And finally, I processed and visualized using RStudio. Sequence typing is a powerful tool to characterize and group bacteria into specific genetic groups. Some sequence types have been shown to be higher risk for clinical spread and severe clinical outcomes, as was previously discussed with sequence types and pandemic lineages. On the right, you see a neighbor joining tree of the sequence types with lineage identification by phylogroup, another form of organization by genetic relationship, and labeled isolate and ST identification. In our study, we found 24 distinct sequence types and 5 unknown sequence types, including pandemic expect lineage ST131, the predominant disease-causing lineage, ST410 and 69, which are associated with greater risk factors for disease and transmission, and numerous ST405s were present, and ST405 is an emerging pathogen seeing global distribution. We also identified ST167s, which are considered emerging ST types associated with carbapenemases. Of note is the phylogroup relationship. Phylogroups have been one of the methods microbiologists and geneticists used to categorize E. coli. Traditionally, extraintestinal pathogens were derived from phylogroups B2 and to a lesser extent D. Yet among all of these really important carbapenem-resistant isolates, they're distributed over a broad range of phylogroups, and that really surprised us. As you recall, FIMH typing is a process of genetic categorization based on the differences in the FIMH gene sequence. And these differences are used to further relate E. coli of the same ST type. On the right, you see a table of the presentation of the phylogroup stratified by FIMH type and ST type, with a given number of isolates and the prevalence belonging to each. Here we see ST131 belongs to global trends of most frequently identifying with H30 subtype. Also of note was S2405, all had the same FIMH subtype, which represents a high degree of relationship. But there also existed a larger variation along the rest of the STs outside of 405. And this indicates a diverse pool of clinically relevant carbapenem-resistant E. coli. In total, 71 acquired resistance genes were identified, representing resistance to 8 classes of antibiotics. Of note is the gene MDFA1, which is a multi-drug resistant gene, which was present in every single isolate. MDFA1 encodes for a broad spectrum of classes of resistance: tetracycline, chloramphenicol, erythromycin, aminoglycosides, and fluoroquinolones. And by itself is worrisome alone from its presence. But as you can imagine, in combination with other resistance genes is even more frightening. We also identified the dominant carbapenemase resistant gene, which was NDM5. And perhaps the most striking characteristic of all of the resistance genes is the total resistance. Among our entire isolate population, the presence of resistance genes to these antibiotic classes was extremely high. Almost 40% of all of our isolates, in all cases, had resistance genes to every single one of these antibiotic classes. As you can also see, some of which are a lot higher. This is particularly worrisome as you think about mediation of disease and infection by antibiotics, as the window of opportunity for treatment greatly narrows. To further characterize the breadth of antimicrobial resistance genes, we felt it essential to relate to sequence type. On the left side of the figure, we have the resistance genes, and on the top, the sequence types, with the dash representing the unknown STs. In the center of the graph is the mean relative abundance of the sequence, the antibiotic resistance sequence, that is. And that's calculated by taking the length of the antimicrobial resistance gene sequence and dividing it by the total length of the isolate gene sequences. And those were averaged to the mean of sequence type. They're also color-coded by antimicrobial resistance class. And the x-axis represents the number of isolates per ST type. Here we see a broad range of carbapenem-resistant genes, but also they're sparsely diverse. Each of these isolates are carbapenem resistant. It was confirmed by the Alameda Department of Public Health. And therefore, the resistance to carbapenems is likely through a combination of chromosomal mutations, as well as acquired non-carbapenemase resistant mechanisms, such as porin and drug efflux pumps. Beta-lactamase genes providing ESBL resistance, such as TEM1B and CTXM15, were also found in most sequence types. There was also a high relative abundance of CTX15, which follows with the literature indicating that the global spread and increase of these genes is highly prevalent. Also of note was the dual presence of the resistance genes OXO1 and CTXM15. It was noted in numerous sequence types, as well as specifically within isolates. A chi-squared test for independence resulted in a significant result with a p-value of less than 0.001, demonstrating a relationship between the combined presence of OXO1 and CTXM15, and it's potentially due to co-location on a plasmid. We also noted high relative abundance of aminoglycosides, and this follows with the previously noted high presence of overall resistance to this antibiotic class. When we look at specific ST types, especially ST131, the dominant disease-causing expect lineage, we see a high relative abundance of carbapenemase resistance genes, including NDM1, as well as aminoglycoside resistance and quinolone resistance. When we look at STs 410, 405, and ST10, we also see an association with severe clinical outcomes from the ST types. And here we see quinolone resistance, macrolide resistance, and several beta-lactamase genes, as well as more aminoglycoside resistance genes. Finally, ST167 had a high relative abundance of NDM5, which follows with recent literature. But also of note was ST73, which had a high relative abundance of NDM5 and is relatively new. As you recall, virulence genes are a gene whose presence or activity is in an organism's genome, is responsible for the pathogenicity of its infective agent. These genes can mediate entry, produce toxins, and allow the organism to adapt to the host environment. On the figure, the left side represents the total number of virulence genes, and the x-axis represents the individual isolates. And the genes are color-coded by extra-intestinal pathogenic virulence genes and non-expect genes. There are a broad set of virulence genes that we looked at from the sample, and some are strongly associated with virulence genes causing extra-intestinal infections like UTIs. Those broad sets, those genes are labeled in blue, while the broad set of genes unassociated are labeled in red. In total, we looked at 560 virulence genes, which were identified within the isolate population, from which you can see each isolate contains numerous virulence genes, but also contains expect virulence genes. Some of these genes help bacteria cause serious disease. On the left of this graph are all the genes associated with an organism being able to cause an extra-intestinal infection, like a UTI or bacteremia. On the top of the graph are the E. coli sequence types, with the dash being a representation of the unknown sequence types. On the bottom is the total number of isolates per sequence type. Again, in the center, but this time with the virulence genes, we see the relative abundance of the genes by virulence type. The virulence type, we have 4 different classifications here, but there are a few more in the literature. There are adhesins, which allow for attachment to host surfaces, siderophore and iron sequestration aerobactins, which allow for iron in low environments like the urinary tract, and protectins, which aid in defense against host and environment, and finally toxins, which directly damage the host and disable immune systems. You can see how these would benefit bacteria in surviving or manipulating their host environment. What we see within our population is that there are 6 common within the isolates from Alameda County. Protectin serum resistance virulence factor KPSM, aerobactin siderophore system IUTA, adhesins PAPA and C, and to a greater extent, FIM-A and FIM-D are all present across many of the sequence types. Again, if we look at a few sequence types we know are associated with clinical outcomes, we see that ST131, 73, and 95 have a variety of virulence factors, including several adhesins, toxins, and for 131, an aerobactin system. And again, we can surmise that part of the reason why these STs are so infectious, while they're able to cause these extraintestinal pathogenic diseases, is because they have multiple tools to manipulate the host environment. Community surveillance, as well as outbreak evaluation through whole genome sequencing, allows the application of both preventative response strategies to emerging community surveillance threats, as well as outbreak identification and relatedness of strains. Vaccine and therapeutic targets based on identified and commonly related genes may provide additional avenues of response to the looming threat of limited antibiotics. Regional prescriptive guidelines with relation to antibiotic resistance and its spread may better inform local clinicians on the threats within their region. However, this research is not yet done. Evaluating the presence of plasmids, their content is pivotal in understanding the mechanisms of horizontal gene transfer. Identifying chromosomal genes compared to plasmid specific genes will help further characterize the relationship to acquisition. We're also in the process of forming a data use agreement to allow us to receive clinical data associated with this genetic information, and being able to relate disease type, location, and other epidemiological factors allows us to generate further meaningful impacts from the data that we already have. And finally, Enterobacteriaceae and resistance to carbapenems in the region is not just limited to Escherichia coli. Further defining the regional resistem is a priority and will help understand both mechanisms of gene transfer and acquisition, as well as just defining the regional environment. Thank you very much for listening to this presentation. It would not have been possible without these amazing people on this slide. Specifically, a massive thanks to my colleagues in the Graham Lab and Dr. Jay Graham. Their guidance and support is beyond words. Also, a huge thanks to the Alameda County Department of Public Health Laboratory. Their feedback and insight on this collaborative effort has been also incredible. I'd also like to thank my wife and my two children, Lily and Braden, for struggling with their long nights as well as just encouraging me through this. Thank you. Have a good night. Or day, wherever you are.